Welcome, everyone. This is Christopher Lee. We're going to be starting in just a minute.
Okay, I have 1 o'clock on the dot. Hello and welcome, everyone. This is Rachel Paul with IAAP, and we are excited to have you joining us today for our Neuro Abilities webinar series kickoff. Before we start today's program, just a few housekeeping items to go over. One, all attendee microphones are muted to prevent any background noise or disruptions. Today's webinar will be recorded and made available on YouTube, and we'll send out information on how you can access that. Closed captioning is being provided today by AI Media. You can select the closed captioning icon on the bottom menu of your screen. And we do have a question and answer box available. We encourage you to post your questions there throughout today's webinar, and we will get to as many as time permits today. And any technical questions or other comments can be left in the chat, and we will try to address those issues as soon as possible. So I'm happy to turn the program over to Christopher Lee to get us started.
Good morning, good afternoon, everyone. This is Christopher Lee. We have a great program for you today. From lab to the real world, restoring movement control and the feeling of artificial limbs and brain implants. It is brought to you by the Patrick McGovern Foundation, NCAM, which is the National Center for Adaptive Neurotechnologies, and IAAP, which is the International Association for Professional Accessibility Professionals, which is a division of the Global Initiative for Inclusive ICT. Again, as I mentioned earlier, my name is Christopher Lee. We have a packed agenda today. I am the Managing Director of IAAP. I'm also the Chief Learning Officer of G3 ICT. I'd like to bring next Patrick McGovern, who's the Chair of the Board of Trustees of the Patrick J. McGovern Foundation. So welcome, Patrick.
Thank you, Christopher. Good morning, good afternoon, and good evening, depending on where you are. Great to see all of you. I'm Patrick McGovern, and I'm delighted to be here representing the Patrick J. McGovern Foundation. I'd like to welcome the Neuro Abilities community tuning in from around the world. I want to begin by recognizing the terrific G3 ICT team behind the Neuro Abilities program, especially Axel Lebois, who spent many years working closely with my father, Pat McGovern, as both president and CEO of IDG Communications and IDC. Today, Axel is a champion of the rights of persons with disabilities in the digital age. Axel, thank you for your leadership and collaboration. The Foundation is honored to be part of this initiative to create a more equitable, inclusive, and accessible world. On behalf of all the trustees, I want to thank the community gathered here today for what promises to be a fascinating conversation. Our foundation carries on the legacy of my father, who had a lifelong fascination with the human brain. He established one of the world's leading neuroscience research institutes at MIT, the McGovern Institute for Brain Research, and he was also years ahead of his time in recognizing that technology can be used for the betterment of humanity. With his trademark optimism, he believed technology could democratize information, accelerate human achievement, and solve our greatest challenges. Neuro Abilities uniquely brings together this cutting edge of neuroscience and technology for the betterment of humanity. It is nothing short of extraordinary to consider the impact this technology can have on future generations. My father was also a master communicator and convener who traveled the world bringing diverse groups of people together. He had a deep commitment for sharing knowledge, insights, and ideas for public good. In that spirit, I believe he would be thrilled to see Neuro Abilities is here creating a space for advocates, neuroscientists, and persons with disabilities to come together to promote research and innovation and prioritize the perspective of those who use these technologies. At the Patrick J. McGovern Foundation, we are carrying the torch of my father's optimism. And one of the most promising frontiers of tech for good is the assistive technology that improves lives and enhances human potential. I'm really excited for today's opportunity to learn from the perspective of a neuroscientist and a user with neurotechnology stands today and the promise it holds for the future. Thank you again for joining us today, and I'm going to pass the virtual mic to Christopher.
All right, thank you, Patrick. I sure appreciate it, and we appreciate your support. So the next speaker up that will be doing some introduction is a colleague and friend of mine, Teresa Bond. She's a research scientist, and she is with NCAM, and she is the chair of the Neuroability Advisory Council. So welcome, Teresa, and take it away.
Thank you, Christopher, and thanks to the IAAP and to the G3 ICT and to the McGovern Foundation, all of whom are making this webinar and Neuroabilities possible. It's really a pleasure to work with the group, and it's really very inspiring to hear that it's carrying on the legacy of the McGovern Foundation, which has such prominence in the neuroscience community. It's my great pleasure to introduce my not so old friends, but people who I've known for some time. Jennifer Collinger, who is a doctor of biomedical engineering. She received her doctorate from the University of Pittsburgh, and she is soon to be an associate professor in the Department of Rehabilitation Neuroengineering laboratory, in the Department of Rehab Medicine. And Jennifer is a very prolific researcher who has been the first author or co-author on over 51 peer-reviewed papers, and she has been a pioneer in the area of restoration of movement to people with severe neuromuscular disorders and spinal cord injury. She is seriously deserving of her new position as an associate professor. And I can tell you that she has been a shining light in this translational field since she was a graduate student. She took enormous initiative, showed enormous initiative, talent, perseverance, and creativity from the very first moment she entered the field. And my own personal experience with Jennifer, besides sharing a position on the board of the BCI Society, where she nurtures an entire community of BCI researchers, was when in 2013, she was the unanimous and outstanding runaway winner of a poster contest that I coordinated for the BCI Society meeting. So she continues in this vein. She's and she has shown remarkable sensitivity to all of the people she works with. One of them being Nathan. Nathan Copeland is a BCI pioneer. He will join us to discuss his experiences as a person who has worked with Jennifer for almost 6 years. Jennifer said 6 years in May. He is one of the only individuals in the world who has been implanted with microelectrodes to enable the movement of a robotic arm, and which I understand he'd like to use to play video games. He's always been interested in engineering, but he has really become a partner in this research with Jennifer and her group, and working sometimes up to 12 hours a week as a colleague. So with that, I'm terribly interested in what these people have to say, and I'm very excited to welcome them.
Well, thank you, Teresa, for that kind introduction, and thank you to Teresa and Christopher for inviting us here to talk about our study and experiences. I'm obviously really excited to be giving this presentation in collaboration with Nathan, because I think that's really a unique perspective to share with the rest of this field is what is it like to be a participant in this study. So I'm going to kick us off with just a little bit of background about our project and some of the scientific goals of it. And then I'll turn it over to Nathan before the question and answer session so that he can talk about some of his favorite memories from the study and things that he would like to share. So our research group is interested in using brain-computer interface technology to try to restore upper limb function. And many of you may be familiar with Kim Anderson's study from 2004, where she asked people with spinal cord injury and specifically quadriplegia, so impairment of the arms and legs, which function, if restored, would have the most impact on your quality of life. And far and away, it was really restoration of arm and hand function that was a top priority for this population. And so we're trying to use brain-computer interfaces, or BCIs, as I might refer to them, to tap into brain activity that still remains intact after spinal cord injury, to bridge that injured spinal cord and restore movement. And there are a lot of ways to record that brain activity. So a lot of you are probably familiar with EEG, so electrodes that sit on the scalp, and those can record from large areas of the brain at the same time to give some information about movement. And then there are also implanted technologies. I'm showing here in this top right picture an electrocorticography or ECOG grid that can lay on the surface of the brain to again record activity from now smaller areas of the brain. The technology that we use in our lab is an intracortical microelectrode array, and you can see a picture of one here on the bottom right. So it's a very small electrode array that has about 100 electrodes on it that can record from single neurons in the brain. And as I mentioned in the title, we're trying to develop a bidirectional BCI. And so what that means is that we have these microelectrode arrays that record activity from motor cortex, shown here in blue. We can decode those signals to try to figure out what the participant is trying to do in terms of their movement and goal. And then at the same time, we typically control a robotic arm. At the same time, we can record from sensors in the robotic arm, translate that information into stimulation patterns that we can then play back through electrodes that are implanted into somatosensory cortex. So if we zoom in at, what information are we getting from these microelectrode arrays? You can see that there are 2 electrode arrays implanted in motor cortex there. If we zoom in on one of those, what each of these black boxes is showing is a recording from a single channel on that electrode array. And if you zoom in at one of those channels, you can actually see if we align these threshold crossings or deviations in voltage, that we're getting activity from here 2 different units. So each of these waveforms is an action potential. And we can look at the way that these action potentials are occurring and the rate at which they're occurring and relate that to movement parameters so that we can give the participants control of the robotic arm. I thought I'd tell you a little bit about how we calibrate the BCI to try to figure out that relationship, because we really want to try to pick up on natural motor commands as opposed to learning to generate commands that can be used for control. And so here's an example showing screenshots from a calibration paradigm that occurs in a virtual environment. And so what we do initially is have the computer control the robotic arm to, first reach to a target, which is this red object in the workspace. Then we give an audio cue to grasp with a particular hand shape. And then there's usually a carry phase, so moving that object in space as you would for many functional tasks, and then a cue to release. And so throughout this calibration, the participant is thinking about moving that virtual arm towards the target to execute those actions. We can record their brain activity and examine the firing rates of all of the neurons that we're recording from. And we can also record the kinematics of the robotic arm. And then essentially we fit a regression model to figure out that relationship. And that's what we use as the basis for control. Now, what's been interesting and shown by many research groups over the years is that this attempted or imagined movement generates activity that is very similar to what is seen during overt or actual reaching movements as has been measured in animals, for example. And so what that means is that this control is fairly intuitive and natural, and that participants typically report thinking about, go grasp that object, as opposed to thinking about extrinsic commands such as move to the right, move a little bit forward. And so when we're recording from populations of neurons, about 200 channels in motor cortex, we've shown that we can enable up to 10-dimensional BCI control, which is 3-dimensional endpoint position, 3-dimensional orientation of the hand in space, and then up to 4 dimensions of hand shape. For most of the work that we do, we typically control about 5 to 7 dimensions, so moving the hand around in space and being able to open and close the hand. And let me show you an example of what performance looks like when Nathan has just visual feedback. So here are his tasks he's performing as part of the Action Research Arm Test. It's commonly used after stroke. And the goal is to pick up these objects and move them as fast as he can towards the platform. And a time of less than 5 seconds is considered able-bodied time. And so he's getting very close to that, even with only visual feedback on a single day. And so you can see again that the control is very natural and fluid as he moves through those different movements. So I told you we were developing a bidirectional BCI, and yet I showed you that the performance was fairly good with only vision. So why do we need somatosensation or sensory feedback at all? Well, for very dexterous and complex tasks, such as buttoning a button, tying your shoes, or brushing your teeth, it's pretty clear that you need sensory feedback to know how to manipulate those objects to perform these fine movements. But we're not quite there yet in terms of BCI control or robotics or FES to reanimate those functions. So even for simpler things like trying to hold a cup level without spilling it and without squeezing it too hard, or passing an object from one person to another, or holding a fragile object, these are tasks that naturally rely on sensory cues in order to maintain the appropriate grasp force, or to figure out the timing of when you need to release an object so that the other person has a hold of it. So let me tell you just a little bit about what it actually feels like and what types of sensations are generated when we stimulate in somatosensory cortex. And I know that Nathan's going to talk about this a little bit more. So as I walk you through this figure on the left, you can see a schematic of the hand. And what we do about once a month is stimulate with set parameters. Here we're using 60 microamps at 100 hertz stimulation for about 1 second. And we ask Nathan to report if he feels something, and if he does, where does he feel it? And he can draw onto the hand the area of that he's feeling. And so what you see on the left is sort of a cumulative summary of areas of the hand that we've been able to generate focal sensations. And they span the index through little finger, typically at the base of the finger. And if you move over to the middle of the figure, I'm showing you a schematic of the arrays as they are implanted in the brain. So the red line there is what's known as the central sulcus, and to the right of it would be sensory cortex. And so you can see that one array on top is more medial towards the midline of the brain, and the bottom one is more lateral towards, out towards the ear. And so what you can see is that when we stimulate through these electrodes, which are, each square is an electrode, we tend to see clustering based on the color. So on the medial array, we're generating sensations from the little finger and then down into the ring and middle finger. And then on the lateral array, we pretty much exclusively generate sensations on the index finger. And this actually lines up well with what is known about the organization of somatosensory cortex, as shown on the right figure, where we would expect to see that organization from thumb most laterally away from midline towards little finger more medially. And so what this means is that even after chronic spinal cord injury, this organization is still preserved. We can tap into this and stimulate and generate sensations that almost immediately feel like they're coming from a particular part of the hand. And this has been very reliable over a period of years. So these sensations can feel like pressure or tapping or tingling. That might depend on the way that we stimulate. Also, if we turn the amplitude up, that can change the intensity of that stimulation. So the last thing that I'll talk about is just what happens when we put these two things together, the motor control and the sensory feedback. And so this is obviously a very collaborative project. It was led by graduate students Charlene Flesher and John Downey, along with our engineer Jeff Weiss and Rob Gaunt, who's leading the sensory part of our study. And so this schematic just shows you, the overall experimental setup, where again, we're recording from motor cortex with those arrays in blue, using that to control the robotic arm in 5-dimensional space, and then taking sensor data from the arm to turn that into sensory feedback in the through stimulation of electrodes and somatosensory cortex. And so the way that we did that was just record from the joint torques from each of the fingers, and then stimulate on an electrode that corresponded to generating a sensation on that finger with larger amplitude for larger joint torques. And I think that the best way to convey the impact that this had on performance is really just to show a video first, where here I'm showing you the fastest trial with sensory feedback on the left, and without sensory feedback on the right from that task that I showed you earlier, the Action Research Arm Test or the ARAT. And so consistently, trials were faster with sensory feedback. And what you see on the right without sensory feedback is that he spends much more time kind of positioning the hand around the object before he feels confident in initiating that grasp. And we saw this for objects of many different sizes and shapes consistently across multiple days. And so I'll leave you with one data slide here. So what I'm showing you here is just a histogram of the amount of time that it took him to complete the trials for that task that I just showed you. So picking up, there's 8 different objects of different sizes and shapes, and he completed those trials multiple times. So across the x-axis on the bottom is time. So how many seconds did it take to complete that task? And on the y-axis is the number of trials that were completed in that time. So shifting this to the left would be faster times. And so you can see here without sensory feedback, his median completion time was about 20 seconds, 21 seconds. And then when we provided ICMS sensory feedback, that median completion time was really cut in half down to 10 seconds. And so importantly, if you pay attention down to this left-hand side of the histogram, 15 of these trials were completed in less than 5 seconds. And I mentioned earlier that on that task, completing a trial in less than 5 seconds is considered an able-bodied performance time. And that was something that we didn't achieve without sensory feedback. So, even though the performance on its face was fairly good with visual feedback, even for a task where he had full vision of the task and was already performing at a high level, sensory feedback was still able to make an immediate and significant improvement. So I hope that I've shown you that a bidirectional BCI can enable a high degree of freedom robotic arm control that allows our participants to perform functional tasks, that we can generate finger-specific tactile percepts in somebody with chronic spinal cord injury, and that putting those two things together improves the ability to perform tasks involving transport of objects when the sensory feedback is provided. So there's a very large team of people that are working on this project, without whom this wouldn't be successful, including our funding from NIH and DARPA. But most importantly, we really couldn't do this without our study participants who dedicate, a lot of time and undergo surgery to allow us to do these studies. And so it's really my great honor to turn the rest of this presentation over to Nathan so that you can hear about his experiences. And then, please feel free to direct your questions to him at the Q&A since this is such a unique opportunity. Okay, Nathan.
Hello, everybody. My name is Nathan Copeland, and in 2004, I was in a car accident that left me a C5 quadriplegic. When I was in rehab in Pittsburgh, they asked me if I wanted to be on a research registry. They said anytime there was someone looking for participants that met certain criteria, they would look at the registry and give me a call. And then fast forward to 2014, I got a call from the team at the University of Pittsburgh about the BCI research, and they asked if that was something I'd be interested in doing, and I thought it was super cool, and I said yes, and that started this going on 6-year adventure. So one of the first tasks that we ended up doing with linking the sensation to the robot was this finger detection task where Rob here is pushing on a finger. The sensor for that finger is telling the computer to stimulate on an electrode that we had already decided or figured out felt like that certain finger, and they blindfolded me, and this is the first time that we did this task, and it worked, I'd say, remarkably well. Even when Rob tries to trick me here and push 2 fingers at once, I still figured it out, even though I was kind of like feeling like, did he really just do that, or am I kind of imagining things? Um, yeah, but so next is the $1,000,000 question that I've been asked a million times, and sometimes it's still kind of hard to explain. What does it feel like? Well, first, sensations are completely electrode dependent. So, you know, uh, you saw that illustration with the layout of uh, which electrode feels like it generates a sensation on which finger. And then depending on uh, what kind of parameters they stimulate at, that sensation can change a lot. So 100 hertz is the pretty standard frequency that they stimulate at, and that can generate sensations uh, like pressure and like a tingle. There's some warmth. You can see what else is in there. A lot of index knuckle finger or knuckle sensations. But then when you change that to like 20 hertz, then I start getting like tapping sensations, start getting weird stuff that I somehow got described as sparkly, which is, kind of thought of it like a firework where it's in like one area, but it's kind of moving around. Kind of, it's one of those things that sometimes they ask me, what it feels like, and I say, I don't know. Why don't you get some brain implants and figure it out? So in 2016, they came to me one day and asked if I wanted to meet the president, and I said, I guess. Like, why would I say no to that? But he was in Pittsburgh for the White House Frontiers Conference, and so I got to meet President Obama and shake his hand with the robot arm and give him a fist bump. And here's a video of that. So for years, this was always what I would describe as the best thing I'd ever gotten to do through this study. But then last spring, I got to fulfill my life's dream of going to Japan. There was uh, a neuroscience conference in Toyama that I got to present a poster at, and I did a presentation at Osaka University and Keio University and a few other places, and I got to have a dream vacation that I thought, after my accident, that I would never get to go there. It's just something I had wanted to do since I was a little kid. And then speaking of things I always wanted to do, when the study first started, they always asked me, what kind of goals I wanted to do, what I would want to do with the implants. And I always said I want to play Final Fantasy 14 with them. And then, it only took 5 years, but I did get to play for a while. I wasn't that good, but it was still fun. I enjoy playing games when I can. Also, my implant day is May 4th, which, if you're a nerd, you know is Star Wars Day, and the lab is full of nerds. So this is how we celebrated one day. I still don't even know whose lightsabers those were, but I ended up coming in that day and they were on the table, and I said, yeah, why wouldn't I want to have a lightsaber battle? One of the funny things about this is that's actually a Luke arm by Deca. So, Luke Skywalker lightsaber battle. So that was pretty fun. Speaking of the Luke arm, that's the second robot arm I've gotten to use, and this is the hardest object in the ARAT test, and this is the first time I've ever completed it. Usually that ends in a bunch of marbles going all over the place and someone on their hands and knees on the floor counting, trying to pick them all up. The third and current robot arm that I'm using right now is a KUKA LBR assembly robot with right hand robotics reflex tactile gripper. This is by far the favorite robot arm that I've got to control with BCI. It is way faster than the others. I really like that it's not anatomical. It allows for things like, as you watch this video, the hand can stay parallel to the table, which lets me work better in the workspace. If I was doing this with the anatomical arm, it's shoulder mounted, and if it moves up, then the hand has to move up and be pointed upwards, because that's how shoulders work. And so that task was just lift up the object and put it on the other side of the table without hitting the middle. That was as many times as I can do it in 2 minutes, and that was my current record of 25. Then another day, another thing that wouldn't be possible with one of the other robotic arms was we were just messing around and seeing if I could put this block on this piece of pipe that is on a spring. That's just stuff that was for another task, and we were just messing around. And so, speaking of other things I've controlled a WAM arm by Barrett. I've used a Glory Ha exoskeleton glove that I used to pick up a taco and eat it. And then in the bottom left there is actually a portable system. It's a tablet and a digital hub that uses different cables than the patient cables, which are the big gray ones. These are just like HDMI cables, but it can do basic cursor tasks. It can't do any of the stimulation stuff, but it is portable. I actually have it at my house now. Since all this pandemic stuff started, I actually got to take it home back in March, and I've been using it to play a bunch of games because that's what I want to do with it. So other things I've done, here's a video of me playing Sonic the Hedgehog. but I also like to just use a drawing program. That's my favorite meal at Taco Bell. Obviously, it's a cheesy gordita crunch and a Baja Blast. You gotta have the fire sauce. I also have a YouTube channel. It's called BCI Can Do Better. That's no spaces. And on that, I have a playlist called My Cyborg Adventure, which is just a collection of study-related videos, news videos, that kind of stuff. So I also always get asked why do it. This is a quote right out of the 23-page consent form. You will not receive any direct benefit from taking part in this research. While it's true that I'm not going to get to keep my implants forever, I'm not going to get a robot arm to take home and all this, I think I've actually received great benefit from taking part in this research. The thing I think I've benefited the most is I've grown as a person. I never would have been able to do these kind of speaking engagements in front of rooms of more than 100 people before. That's something I kind of just have grown into enjoying. Really like hearing people say they've found inspiration or value in hearing my experiences, but more importantly, why I decided to do it was to push the science forward. I knew the participants they were looking for is very specific criteria, and I met that criteria, and somebody had to do it. So how could I say no if I could do it? So I want to do whatever I can to make sure that people in situations like mine in the future kind of bypass that that realization that hits all of us that have a catastrophic injury, where we realize our life will never be the same, and we kind of go through this despair of feeling like we're never gonna get to do the things we enjoy anymore, or contribute to society, or that kind of thing. So I'm hoping that the science can get pushed forward thanks to whatever I can do through this study. And maybe one day it really ends up being like Star Wars, where Luke got his hand chopped off and the next day he had a robotic one that could feel a pinprick and worked just like his the hand he had before, and was good enough to save the universe or whatever. So yeah, thank you guys for listening to what I have to say. That's me in Japan at a park with lots of deer. Okay, so yeah, I guess now is the time for questions.
Thank you, Nathan. Thank you, Jennifer. We appreciate the incredible work and incredible story. Rachel, let's open it up for questions. I know we have a few in the Q&A as well as the chat.
Yes, this is Rachel. I'm going to get started because we do have quite a few. So the first question might be more directed towards Jennifer, asking, is Nathan using only his somatosensory, I don't know if I'm saying that correct, cortex, or is he simultaneously using his motor cortex to move and feel?
So for almost all of our experiments, we decode the movement signal from motor cortex and then stimulate in somatosensory cortex for the sensory feedback.
Okay. Our next question, congratulations, Nathan and the team. Can you speak about the muscular capacity of the robotic arm? Like what weights can the arm lift? And does functionality cover the whole arm or part of it or just the hands?
Yeah, so the KUKA robot, the one that Nathan likes, is the strongest, I would say. I think it can hold 14 kilograms. So weight is not really a concern. And, we don't even really need to deal with the effect of gravity. So the way that we've controlled all of these robots is really in sort of endpoint space. So if you think about what Nathan's trying to control is to position the hand in the place and orientation that he wants it to be. And so the computer figures out how to control all of the joints to get it to that position. But what we're decoding from the brain is the endpoint velocity that essentially sets the position and orientation of the hand.
Our next question, I noticed Nathan has some use of his hands. Did this experiment help his brain to recover that use?
No, I don't know, Nathan, do you want to talk about your injury level and how much function you have?
Okay, so I'm C5 quadriplegic. So I do have some wrist function. I have wrist extension, but not flexion. And I have no finger movements at all. And then I have pretty good arm movements, biceps, no triceps, but good shoulder, all that. And no, unfortunately, none of this has the capacity to, regain any of my lost function. But it'd be cool if it did.
Yeah, I would just add to that, we've been using robots for control, not necessarily because we think that's the ultimate device that people want, but because we're trying to focus on how do we get this information out of the brain for control and then use stimulation to provide that sensory feedback back. And so robots are very reliable in terms of, going where you tell them to. There are definitely other groups that are working on trying to use electrical stimulation of the nerves or muscles to restore function to people's own limbs. And that's a very challenging but important problem that people are working on that, might have the capacity for some functional recovery, and more closely align, I think, with what people are desiring.
Okay. Just wanted to mention we have a question from Mohammed, and I'm going to hold off since you're asking about some other projects and specifically related to optic nerve or vision. So if we have time at the end, we'll come back to that, or maybe I can put you in touch with Jennifer afterwards. So I just want to keep questions for Nathan here. Nathan, I know you talked about this a little bit. Do you find certain robots to be easier to learn and or control than others?
So I'd say controlling them is not more difficult from one robot to the next, but okay, so like Jen said, control is endpoint, and the robot kind of figures out what to do to get the hand there. And so each robot moves within the workspace differently. They have different kinematics. And so that's the main reason that the Luke arm is my least favorite arm that I've controlled, because just how it was more limited in the workspace. Like if you would reach to the far left, it would, bend at the elbow a certain way, and it just kind of was more limiting than, say, the KUKA. Like I mentioned, when I'm doing a task, I can move the hand part in anywhere within the workspace because it has so many joints that it can stay parallel to the table. So that's why I think an anatomical robot, say, you know, years down the road, there's one that can get mounted in a house, and you want to use it in the kitchen, and you want to get a box off the shelf in the cupboard. If you're using an anatomical arm that's mounted at the shoulder and you need to reach the top shelf, when you move the arm up, it's going to bend at the elbow, it's going to put the hand at a position that's pointing towards the ceiling, and you won't be able to grab anything off the shelf because your hand's going to be pointed up. But with the KUKA, I could move up and it would stay parallel to the ground, and I could grab the box and pull it off and bring it down. But like you asked, the control is not harder. Learning to use or recognize how they work within the workspace has been different with each arm.
Okay. Thank you, Nathan. We have more questions here for Jennifer. Did the arm get calibrated each time it was used?
Yeah, for almost all of our experiments, we train a new, we call decoder, so figuring out that map between brain activity and kinematics each day. It's the same general relationship, we assume, but the neurons that we're recording from can be different from day to day.
Okay. Another question for Nathan. You shared My Cyborg Adventure. Is that something that is shared with the general public if people wanted to follow you or find that?
Yeah, so the actual YouTube channel is called BCI Can Do Better. That's no spaces. But if you search on YouTube for My Cyborg Adventure and filter by playlist, it should come up.
Okay, great. Let's see, I think we have a few more questions. So let's see. So it looks like you have functional use of your right arm, and how will you decide which to use for the robotic?
Yeah, well, it started out with, we had a right-handed robot initially, and so we were planning to enroll someone and do a left-sided implant, right? Because the left side of your brain typically controls your right arm. There's been some discussion about whether, if somebody was left-handed prior to their injury, would we still get as strong of a control signal from their non-dominant hemisphere? And fortunately, we haven't had to debate that yet because all of our participants were previously right-handed prior to their injury, but also significantly impaired after injury. And so it was kind of a mix of practical reasons and then just thinking that the dominant hemisphere would have the strongest, most coordinated command signals, but we really haven't tested that.
Okay, another question for Nathan. It says, did you experience any training effects in how easy it is to try to generate the movements necessary to control the robotic arm?
No, like it's just been like, I guess, really intuitive. Like I never really had to like go through this whole trying to learn how to think something to make it move right, other than just moving, think about moving my arm to the right. And so it's one of those things that it worked the first time we tried it. Of course, we start with only like 2 degrees of freedom. Like I move the arm up and down and left and right, and it worked. It was just, I watched the computer move into the targets, and then I just think about moving my arm in the same way, and then it trains the decoder, and it just works. And yes, so that's all I've ever had to think to get it to move. Of course, you start slow and you do the 2 degrees, then you add the third so you can move it in and out, and then grasp and wrist rotations and that kind of stuff. But yeah, no real mental gymnastics or struggles.
I think on the same lines, another question was, has your quality of life improved with the device being at home? I know you mentioned you were able to had a portable device you were able to bring home.
Yeah, like for a while there, I was using it like almost every day because the world shut down and I couldn't go anywhere, and so that kind of definitely saved me from some of the boredom and driving myself crazy with nothing to do. So I mostly played games, and I drew a cat, and I should have put it on here too, but yeah, I spent hours working on this cat. And yeah, so at home the system's pretty much limited to that. I can train a decoder that can move a cursor, and I can play games or draw. So it's fun. I want to get back into using it more often and play more games. And new consoles came out, and maybe get one of those and control a PS5 with my brain, and I want to get a nice new TV and computer, and I don't know, send some emails out to companies and see if they think I'm cool enough to give me a TV and a Xbox and a PlayStation.
Okay, we have a few more questions here. Did you ever experience any sensations from the stimulation other than the ones in the robotic arm?
I think kind of what this question's asking is, have I received sensations that were not from tasks using the robotic arm? The answer is yeah. There's all kinds of tasks that use stimulation, but not involving the robotic arm at all. But also, what they also might have meant is, no, I've never felt any sensations just every day not hooked up to the arm and receiving stimulation.
Most of the tasks rely on the fact that stimulation through a given electrode generates a sensation that feels like it's coming from somewhere on Nathan's hand. And so it could be driven by virtual reality or some other mapping that we provide. But Nathan, correct me if I'm wrong, but it always feels like somewhere on your hand. It's not an abstract sensation.
No, always my hand.
Okay, we have a few more minutes here. I think we have 1 or 2 more questions. How has this BCI collaboration benefited other study participants and the academic curriculum at the university?
I think this is the only implanted BCI study that we have going on here, although we have expanded to now a site at Chicago as well as part of this study. And so I think that's a very small community of people who are doing this type of research. And that's one of the reasons that we're interested in doing presentations like this, is to share our experience and what it has been like. Certainly, we've collaborated a lot with basic neuroscientists who, have always wanted to ask, what it feels like to receive this type of stimulation, have been unable to in animals, for example. And as Nathan explained, it's still maybe a little bit hard to articulate in words, but it's been just a great collaboration with other neuroscientists in this area.
And piggybacking off of that, I wanted to go back to Mohammed's question, since we have a little time, and this could be open up to the entire panel here, but he was asking, to what extent have other areas of BCI tests have been as successful in comparison to the robotic arm? Has there been any tests for sensory-related BCI, especially with a regard to the optic nerve or vision-related functions in general?
Yeah, I mean, on the motor control side, I think there's been a lot of great progress over the past few years, just in terms of communication. So trying to decode either cursor control for typing on a virtual keyboard, or even recent approaches of decoding handwriting, for example, that have really accelerated the pace that can be achieved for BCI-driven communication. And I mentioned earlier that other groups are trying to tie BCI with functional electrical stimulation, either for assistive or rehabilitative purposes, which I think is exciting. The sensory BCI in terms of vision, I don't know if there's other panelists who want to speak to this, since it's a little bit outside of my area, but definitely there are cortical visual prosthetics, that are now, FDA cleared and under investigational trials for restoring vision. The general safety principles and things are similar, but the science is different.
Okay. And then another question for Jennifer. What is your impression of the recent interest of the private sector in BCI development?
Yeah, I mean, one thing that I think is challenging for this field is, how do we actually translate it out of the lab, and so that people can use it at home, and not just for experiments or playing video games like Nathan mentioned, with our portable system, but actually on a big scale of translating this and making it accessible. And that's going to take significant investment. And so I'm personally excited to see the interest in this technology, to try to push forward the actual implants and electronic technology. It's clearly going to need push in terms of science, so that we can develop technology that's reliable and robust outside of the lab. And so, I think there's going to have to be tight integration with academia, and then figuring out actually how do we get this out as a medical product and reimbursable for people to be able to use. So, I do think that this investment is important, and it's part of the responsibility of this community to try to provide input into that, to make sure that it gets back to helping the people that we're trying to help.
Now this is Christopher. We have one more question, and I can't help but to ask it. Nathan, you mentioned earlier about pushing science forward, and that's what it's all about for you. It seems like. I'm just curious, where's the science going? What's next? Nathan or Jennifer, either one.
I mean, it's hard to know. I think every time something comes out, and you look back 6 years ago, and I never would have thought I could be doing the things that I'm doing now. So in another 5 years, I really don't know, but hopefully, like Jen said, there's pushes by people with money that want to make things, and that's one of the things, like, okay, if we get the implants and the that stuff working out well enough that you can go home with it, you also need stuff to control, like I mentioned a KUKA in the kitchen. That would never work. That thing's huge, and so there needs to be people thinking about stuff like that. Like, what are you gonna do with these implants? Like, you need a KUKA that is, mini, like a mini that could be in your house, could attach to your wheelchair. That kind of stuff will come down the line eventually, and everyone can play more games.
Well, I just wanted to add that, it's because of you, in part because of your courage to try something new, and Jennifer's courage to encourage the science and to follow the science in this way, that private industry is interested, and that the science is moving forward. So I have to say thank you to you and to Jennifer.
Thank you. Our time has ended. To the presenters, awesome presentation. I want to thank you all, and I want to thank AI Media for being our captioning team today. I want to thank G3ICT and the team. We do have our next webinar coming up in January, so stay connected. Make sure you check us out, and we look forward to getting more information out to you. Thank you, everyone.
Thank you.
Thank you.
Thanks.
Bye-bye.
Bye.
Thanks, Jennifer and Nathan.